Hello, my name is Connor Janetto Hill, and my poster is on designing chemostat media to replicate the diets of hunter-gatherer communities. So let's start by talking about the gut microbiome. It's one of the most diverse microbial communities found on the human body and is located in the large intestine. It has many influences in health and diseases such as metabolic diseases and inflammatory bowel diseases, and generally, more diverse is better. I also want to define two different lifestyles. So the first is what we're most familiar with. It's the industrialized lifestyle. And it's characterized by having access to farmed and processed foods along with medicines such as antibiotics. This is contrasted to the traditional lifestyle, which is characterized by a more hunter-gatherer based lifestyle. And it's been noticed that the traditional lifestyle gut microbiome is more diverse and contains species not found in the industrialized gut microbiome. And rates of diseases associated with the gut microbiome, such as inflammatory bowel disease and metabolic diseases, are lower in traditional populations. This has led to the creation of the ancestral microbes hypothesis, which states that the traditional lifestyle has limited changes compared to ancestral humans. This has allowed for the retention of what are called ancestral microbes. And these ancestral microbes are what prevents the metabolic and inflammatory diseases. Compared to the industrialized lifestyle, which is highly changed compared to ancestral humans, and has allowed for the loss of these ancestral microbes. And this has increased the risk of metabolic and inflammatory disease rates. A group living a traditional lifestyle are the Yanomami, and they live in the Amazon rainforest with diets higher in carbohydrates and lower in fats and proteins compared to an industrialized lifestyle. And these dietary differences could account for the increase in microbial diversity, along with the presence of certain microbes that aren't found in the industrialized gut. And these novel microbial species could be these ancestral microorganisms. So it is my hypothesis that growing a defined community in vitro in a media composition that replicates the Yanomami diet can produce a different community structure compared to an industrialized diet formulation. To test this, I'm going to be growing a community derived from an industrialized donor in a chemostat system, which I will explain in a minute, using new formulations to replicate the Yanomami diet. I will then take these communities and compare them using Illumina sequencing. So, a chemostat, or a robogut system, replicates the conditions found in the large intestine. This means maintaining the pH at a pH of 7, constantly having nutrients flow in and out, maintaining a temperature of 37 degrees, and most importantly, keeping the system anaerobic through the use of nitrogen gas. Table 1 indicates the different media compositions between each of the media types. And as you can see, the YAN1 and YAN2 media formulation contain different proportions of starch and protein compared to the industrialized diet formulation. And between themselves, the protein source is different. Below, in figure 2, you can see the experimental flowchart. And what we did is we took an industrialized gut-defined bacterial community and inoculated 3 chemostats containing each of the 3 media types. This was run for 16 days at the conditions stated above, with a sample being taken every day. And every other day's sample was sent for Illumina sequencing. Now for the results. So the community composition throughout the chemostat runs is shown in figure 3, with the 25 most abundant species being presented. The top microbial species between the 3 conditions are roughly the same. However, their abundances are what is different. And comparing each of these community compositions using beta diversity Bay-Curtis index, these 3 communities differ on the species and genus level. This result is also echoed in figure 5, showing the Yanomami diet formulation 1 and Yanomami diet formulation 2 differ from the industrialized diet formulation independently. For a more detailed look at the community differences, genera abundances were examined. It was found that Yanomami diet formulation 1 and 2 shared some genera that were different from the industrialized diet formulation community. Genera differences were also observed when looking at the YAN1 community on its own, observing that Corpococcus and Eubacterium were lower compared to the industrialized diet. In figure 8, the YAN2 community was higher in abundance of Doria compared to the industrialized diet. What these results show us is that the different media formulations resulted in different community structures, and is evidenced by figure 3, The Yanomami diet formulations are closer to each other than they are to the industrialized diet community. Differences, however, do exist between the two Yanomami diet formulation communities. This proves that the differences in the Yanomami diet formulations were enough to create distinct communities of bacteria, even when using the same starting inoculum. In the future, we would like to run these samples on nuclear magnetic resonance, or NMR, to see what metabolites are present to understand what the bacteria were doing, and looking at the metabolic output of each community and see how they differ. We will also be using this Chemostat media to replicate the Yanomami diet in hopes of growing Yanomami stool and isolating some of these microbes that are only found in traditional population guts. It is our end goal to create a defined community that is representative of true Yanomami gut samples and to allow for reproducible experiments in the Chemostat.